faba, fab, faba beans, horse bean, windsor bean are simply al-ful al-Masri, and many other names that are given to one of the most ancient and popular legume in the world. In fact, probably one of you had it for breakfast today. Yes, I'm talking about the Vicia faba. Hello and good day for all of you ladies and gentlemen. My name is Howyda Hashim. I am a graduate of Pharmacy School, University of Medical Sciences and Technology, Sudan. First, I'd like to apologize for my absence, but this was due to some personal circumstances that I couldn't manage. If you may allow me to take around 20 minutes of your time to share with you this information about this research under the title Evaluation of the Nephroprotective Activity of Vicia faba against Gentamicin induced nephrotoxicity. I'll be talking about herbal medicine, acute kidney injury, a brief account on the Gentamicin induced nephrotoxicity and Vicia faba. I'll also share with you the objective of this research and why I chose to work in it. Materials, methods, and results will all be discussed, and I'm going to start with the conclusion of this research and the findings, in addition to some recommendations. Of course, whatever I say in this research or in this presentation is cited, with the exception of the findings and the recommendations. So let's get started. Herbal medicine, botanical medicine, or phytomedicine, they all refer to a branch of science that uses a whole part or extract of plant for medicinal purposes, whether for antagonistic, treatment, or even prophylactic. Of course, this information is not for all of you. Plants have been playing an important role since ever, even before recorded history, not just for their nutritional value or for their medical uses, but they were used in clothing, fuel, beverages, dyes, cosmetics, and many others. There are many drugs that have been derived directly or indirectly from plants, such as the aspirin, morphine, atropine. In fact, 11% of 252 drugs were considered as basic and essential by the WHO, were all derived from plant origin. But unfortunately, herbal medicine has its limitations. Why? Because the herbal remedies, after all, they are natural products. They differ in their chemical constitution according to various factors, such as the type, the part of the plant used, the climate, where this plant was cultivated, whether it was cultivated in Sudan, cultivated in Egypt, cultivated in South Europe, in South America, or any other part of the world. All these factors are going to affect the chemical composition. Affecting the chemical composition will definitely affect the pharmacological activity, whether it's going to decrease it, increase it, or even abolish that activity. Acute kidney injury. It's known for all of us that an abrupt or rapid decline in the renal filtration function associated with an increase in the creatinine, in the levels of the creatinine, is known as acute kidney injury. There are a number of precipitating factors that includes dehydration, obstruction of urine flow, sepsis, surgeries, or even drugs. Drugs such as the non-steroidal anti-inflammatory drugs, antibiotics, gentamicin, which I'm concerned with in this research, cisplatin. Unfortunately, acute kidney injury has no specific treatment. There is no drug X can be used for this AKI. The treatment of the AKI depends on the underlying cause. For example, if it was dehydration, then I should rehydrate my patient. If the problem was a medication, then I should stop that medication. But stopping the medication usually is followed by negative consequences, such as abrupt stopping of antibiotic therapy. We all know that abrupt stopping of antibiotic is one of the causes of the emerging antibiotic resistance. Okay, gentamicin. Gentamicin belongs to the aminoglycosides, a class of antibiotics that mainly acts on the gram-negative bacteria, in addition to some Pseudomonas species. But aminoglycosides, they are known for their nephrotoxicity, neurotoxicity, and ototoxicity. All affect the dosing of these drugs. I'll be speaking mainly on gentamicin. Gentamicin has a pronounced nephrotoxicity, and that is done by increasing the oxidative stress. How does it increase the oxidative stress? By several pathological pathways, whether increasing the endothelin 1, increasing upregulation of the cross factor beta 1, infiltration of monocytes and macrophages. All these lead to increase in the free radical production and its accumulation in the nephrons, specifically in the glomeruli, and causing the nephrotoxicity. So I'm going to talk now about the Vicia faba. Vicia faba, a part of its tasty, being it tasty as a food, but it's one of the ancient and annual legumes in the world. It belongs to the Fabaceae family. It's believed to be native to North Africa and Southwest Asia, but it can be cultivated anywhere. The faba seeds, they are rich in many secondary metabolites, mainly fatty acids such as the phytosterols. And we all know the importance of phytosterols as antioxidants or in lowering the cholesterol levels. Also, the faba leaves are very rich in secondary metabolites. Almost all secondary metabolites, alkaloids, flavonoids, terpenoids, tannins, they're all present in the faba leaves. Faba beans were traditionally used just as a nutrition, not just as nutrition, but they had aphrodisiac activity in addition to a potential use in Parkinson's disease. Both are due to the fact that faba beans are rich in L-dopamine. Faba leaves also possesses muscle relaxant activity, besides its traditional use in treating kidney stones and eye infections. What was the objective of this research? Was to perform phytochemical screening, preliminary phytochemical screening of the dried leaves of the faba leaves, to evaluate the nephroprotective activity of Vicia faba extracted by two different methods on gentamicin-induced nephrotoxicity in rats. Why I did this research? 19 to 33% of the in-hospital AKIs were reported due to drug nephrotoxicity, and one of the most cited drugs was gentamicin. The only management, as I mentioned earlier, was managing the underlying cause, managing the therapeutic biochemical abnormalities and maintenance of the volume homeostasis, or stopping the medication until the AKI resolves can reverse the damage, but this will definitely affect the outcome of the antibiotic therapy. The plant material used was in its shed, dried, fine powdered form. The extraction of the plant material was done by two different methods: the succulent extraction or the continuous extraction using 96% ethanol, and decoction extraction using distilled water. The phytochemical screening was done according to the Musa al-Hadi standard qualitative method to detect the presence of various secondary metabolites, while the pharmacological experiment was adapted from a research article by Ender Hor et al. Published in 2013. The induction of the nephrotoxicity was done by the gentamicin, and the standard drug was the cystone, which is a non-nephroprotective herbal remedy. And I had two test groups. One was the ethanolic and the other was the aqueous extract, both with the same dose, 500 milligram per kilogram. The results. I divided the results into three: the phytochemical screening, the pharmacological activity, and the histopathology done for the kidneys. From this slide, I don't know if it's very obvious for you, but in this slide you can see that almost all the secondary metabolites, the alkaloids, the saponins, tannins, anthraquinones, cardiac glycosides, and furogens were all present in the faba leaves. And now, let us move to the nephroprotective activity, which I'm concerned with. I would like to share with you the magnificent effect of the ethanolic extract in reducing the creatinine levels by a highly significant value, even more than the positive control. It also had a very significant effect in reduction of the urea, and it increased the albumin levels. Yes, it had no effect on the total protein, but the effect of the ethanolic extract was superior to that of the positive control and superior to that of the aqueous extract. The aqueous extract had an effect on the creatinine, but not as much as the ethanolic. A reduction in the creatinine, but not as much as the ethanolic. Also, it has a reduction in the urea and an increase in the albumin levels. It also didn't have any effect on the total proteins. This graph will elaborate my result more easier and make it more easier to interpret the result. If you noticed here, let us take for example, the urea. The reduction in the urea by the ethanolic extract was 53.6, while by the positive control, the system was 42.8. But if we went back, we find that we're going to find that the standard deviation of the ethanolic extract in reducing the urea was just 1.14, while that of the positive control was 13.42. Very light deviation from the mean. That's why the ethanolic extract was superior to the positive control and superior to the aqueous extract. If we can also take a look at the albumin, we find that the albumin in the ethanolic extract was almost the same as the normal. Like in the ethanolic extract, it was 3.92, while in the normal group, it was 3.98. Almost the same. We find that the aqueous extract follows directly the ethanolic extract in increasing the albumin levels by up to 3.8. Now let us move to the histological examination. These are slides from all rats. I combined them together for the kidneys. Okay, this histopathology the comment on this histopathology was done by an expert in histopathology, Professor Abdurrahman Jamil. He commented on the slides. He said that in the negative control, he found that the cell swelling, especially in proximal tubules, with multiple foci and mononucleate cell infiltration, in addition to the presence of large amount of tubular casts. Of course, all these features are definitely a characteristic feature of AKI. If we move to the positive control, we find that these features, the tubular casts, the infiltrations, and the tufts were less in the positive control, in the group that was taking the cystone. We also going to find in the next slide, which is this slide is for the ethanolic extract. We're going to find that the kidney chains are almost the same as that of the positive control. Surprisingly, the aqueous extract had the best effect on the kidneys. Less swelling, almost no casts, almost no infiltration. This result was to some extent surprising for me, because when I compared it to the biochemical markers, the ethanolic extract had a better effect in reducing the creatinine and the urea and increasing the albumin, followed by the aqueous extract, followed by the positive. So the histological examination revealed another result, that the aqueous extract had a better effect in protecting the cell. In conclusion, fava beans are definitely enriched with secondary metabolites, and we all know the importance of the secondary metabolites, whether the antioxidant effects of flavonoids, the antimicrobial effects of tannins, or the effects of the alkaloids. They're all important. I also concluded from the result that definitely the ethanolic and the aqueous extract have a nephroprotective activity. But in terms of the biochemical markers, the ethanolic extract was superior to the aqueous extract. This might be probably that the ethanolic extract usually is the crude extract. So probably there are compounds that are present in the ethanolic group and they're not present in the aqueous extract. also the histological examination, the aqueous extract exhibited a better effect regarding the reduction of the tubular casts than the ethanolic, than the positive control. Finally, I would like to recommend that this experiment or this research should be done on a larger scale, with including more number of rats, of course to omit the individual variations. In addition to that, I mentioned earlier that the gentamicin increases the oxidative stress in the kidneys. So it would be better if we are going to monitor the catalase, glutathione oxidase and lipid peroxidase to have a clear picture on the effects of the extract. Not just the biochemical markers, we should take a look also on we should also monitor the catalase and the glutathione and these enzymes. I recommend also that the crude extract of the Vicia faba should be subjected to HPLC, MS. Why? To have a full identification to the phytochemicals present in these leaves. Evaluating the biological activity using fractional extraction. Why? To identify which fraction has the activity the most activity, and that will help to determine the active phytochemical behind its activity and the exact mechanism of action. This is the references for the introduction and for the Vicia faba, for the method and for the materials I used. If there is any questions or comments, please do not hesitate to email me at howyda96@gmail.com, but please make them positive comments. Thank you for listening and goodbye.